I'm David Rowich, and my new fellows talk is entitled Heterogeneous Functions of Glia. This image of a brain embedded in the cosmos is apt because one of the cells that I'll be speaking about is the astrocyte, so called because of its star-like appearance. Now, glia have been present during evolution for eons, going way back to primitive invertebrates. But as we track the percentage of glial cells comprising total cells in the brain, they've increased to 90% in the human brain, which implies they've had important functions enabling more complex brain activity. And one specific example in evolutionary innovation occurs at the transition from jawless to jawed fishes when we start to see myelinating glia, oligodendrocytes, and Schwann cells. And the cartoon shows the difference in nervous impulse transmission with myelin on the right, much faster, with much smaller axons, allowing for greater packing, greater number of neurons, and therefore greater brain complexity. So my lab has been interested in development of glia in new roles. They are 90% of cells in the human brain, and this is in contrast to neurons, but they haven't achieved quite the same amount of attention as neurons over the last 150 years, possibly because of an unfortunate name, glia, which means nerve glue, the sort of substance sticking together neurons. So they're relatively under-researched. Astrocytes represent total of 50% of cells in the brain, and they're ubiquitous and could be determinants of many local environments. So are they heterogeneous? And oligodendrocytes, which have classic roles in myelination, but might have other roles. And this is where my lab has used developmental tools to try to address some questions. Now, when my lab started this investigation over 20 years ago, the traditional view was that glia are the sort of gray-suited cells of the nervous system, homogeneous with common origins. But can we use that developmental and genetic approach to ask, can we determine features of heterogeneity? And generally, our approaches have been to use functional screens to determine genes that could be specific for glial subtypes, or conversely, use gene expression as a clue to function, which we would then validate, typically in mammalian systems. An overriding hypothesis has been that pattern formation of the central nervous system regulates glial diversification and function. And one great example of a gene that has a critical role in glial development is OLIG1 and OLIG2 gene family. This is expressed in a very tiny strip within the spinal cord, which you can see in this sort of circa 1990 spatial transcriptoma called Mount situate hybridization. And at higher power in the panel, you can see that OLIG proteins are expressed in a very small segmental region of the ventral spinal cord. And this was a clue that the origins of oligodendrocytes could be distinct from other types of glia, and allowed us to follow this up with functional studies. Work carried out when my lab opened at Dana-Farber Cancer Institute with Chuck Stiles and postdoc Richard Liu. And now we know that OLIG2 is a critical and the first transcription factor actually essential for development of a glial subtype, the oligodendrocyte. It's expressed in multipotent precursor cells, where it is required for the specification of the lineage, and is required also at all stages of the lineage described in the cartoon as they migrate throughout the central nervous system, find axonal targets, and then in the lower right, perform their myelinating function, which is so critical for normal neurological activity. And of course, can be involved in diseases like multiple sclerosis, where those cells are attacked. Now, a further clue about the diversification of glial subtypes came from collaborative work that we did with Stu Orkin's lab, where we found that the BHLH transcription factor SCL or TAL1 was expressed in an adjacent domain to OLIG2 and participated in cross-antagonistic interactions such that if SCL was dominant, astrocytes would be produced versus OLIG2 as a domain that enhanced oligodendrocyte development. So that began to develop the premise that pattern formation, that restricted origins of glial subtypes could be related. We followed these studies up in collaboration with work done with Bill Richardson at UCL, in which through exhaustive fate mapping of multiple domains in the spinal cord and brain, found that these progenitor domains highlighted in different colors that are determined through patterning and the impact of morphogens like BMPs and Wnt dorsally or sonic hedgehog ventrally, result in progenitor domains that would give rise to astrocytes that were restricted in their allocation to specific dorsal ventral or anterior posterior locations from which they emerged, so that we could start to now trace an embryonic origin during development to an ultimate astrocyte cell fate, even in the adult animal, suggesting the possibility that functional specification and diversification could be programmed at a very early stage. And to look into that, we did some simple experiments, like comparing the astrocytes from dorsal spinal cord to ventral spinal cord. And that identified one very strongly expressed ventral gene in astrocytes called SEMA3A, which is a member of the somaphrin family involved in axon guidance. And shown on the left-hand sort of cartoon, you can see this somaphrin 3A expression in turquoise, occupying astrocytes in the ventral half of the spinal cord. You also see two neuron types, alpha motor neurons and gamma motor neurons highlighted in orange. And that shows the normal distribution of those types on the left. In contrast, on the right, the SEMA3A conditional knockout, where we just removed it from astrocytes of ventral spinal cord, showed several abnormalities. First, misguidance of axons that were penetrating to ventral from dorsal, but also importantly, we lost about 50% of fast alpha motor neurons. So this was a novel function for SEMA3A encoded by astrocytes to maintain a sort of trophic and supportive role for astrocytes. And again, was one of the first examples of how functional specification of astrocytes in region-restricted domains could have important roles in the maintenance of neuronal populations. Now, if you lose your fast alpha motor neurons, that predicts a phenotype which was confirmed by Anna Malewski in the lab, where they have significantly decreased peak strength. And so this is not a mouse that's training to be in the circus. He's trying to hold on, or she's trying to hold on. And we're measuring peak strength using this and other measures. And so when you lose your fast alpha motor neurons, you've lost peak strength. And so that's another example of an evolutionary advantage of a specialized astrocyte of the ventral spinal cord, because you can imagine that having peak strength or being stronger could be decisive in a fight or flight or battle with a competitor. We found a similar finding with another ventral astrocyte restricted gene called KIR4.1. Again, knockout in that scenario also results in loss of peak strength. And so again, suggesting this idea that the local domains determined by region-restricted astrocyte functions can be important for neurological performance. Well, those were examples I gave you from the spinal cord. And of course, now we're using novel techniques like spatial transcriptomics, single cell and single nucleus approaches to now look for greater evidence of astrocyte diversity in cortex. On the left shows a RNA-scope image where we've labeled different neuronal populations, and you can see the beautiful layered patterns that occur. And we're now finding in work from former postdoc Omer Bayraktar that astrocytes also have layer-specific expression patterns, and now we'll be delving into their functions where we expect they may also have roles in the maintenance of different cortical neuron subtype activities. So are glia heterogeneous in 2000? This was a question. We could say yes in 2021, but this is not just a sort of a Photoshop trick. Importantly, this has given us insights into how glial heterogeneity is essential for brain complexity and regional specifications, such as the regional centers of the brain, which we think are imbued with specialized astrocytes to support those functions. The communications between the brain, which obviously require the myelinated functions of oligodendrocytes. And that also applies to connections between the spinal cord and the soma through enhanced conduction provided by Schwann cell myelination. And finally, I've given you an example of how local circuit regulation by the ventrally specialized astrocytes expressing SEMA3A or KIR4.1 are critical for support of key neurological functions. So this gives us, I think, some new and fundamental insights about how our nervous system can function optimally, perhaps how it was selected for optimal function over time. But then importantly, this also overlaps with my role as a pediatrician taking care of children with neurological problems. How can astrocyte or oligodendrocyte or glial-based neurological diseases be better explained? And so this work has set up a framework for further investigation. Thanks very much for your attention.